All right, so note that it's important that you need 6 ATP and 6 NADPH molecules are used to convert these 6 molecules of PGA to 6 molecules of PGAL, also known as G3P. So kind of like what you've been noticing here is that if you have a total of 6 of these 3 phosphoglycerates, then you're going to need 6 ATP. If you have 6 to make it into 1,3-bis phosphoglycerate. Same thing, if you need to convert 1,3-bis phosphoglycerate into glyceraldehyde, 3-phosphate, then you're going to need a total of 6 NADPH if there's a total of 6 molecules. So it's a 1-to-1 ratio for this process here that's taking place. Okay, and at this point here, what happens between the end of phase 2 and the beginning of phase 3 is that you end up with a total of, again, if you're using 3 carbon dioxides, you end up with a total of 6 glyceraldehyde 3-phosphate, G3P. And what happens is that one of these 6 molecules actually leave, and this G3P is going to leave and go into another metabolic process where it's actually going to go to be able to create glucose. However, we need 2 of the glyceraldehyde 3-phosphate in order to make 1 glucose molecule. So if every time you go through this process and only 1 out of the 6 leave in order to create half of the glucose, then you're going to need to go through this process again. You're going to need another total of 3 carbon dioxide molecules in order to allow for 1 of the 6 glyceraldehyde 3-phosphate to leave. So that's why during the video I kept saying that you need a total of 6 carbon dioxides and then you end up with 12 of these products, because if you end up with a total of 12 glyceraldehyde 3-phosphate G3P, then 2 out of the 12 G3P is going to leave, and those 2 G3Ps are going to go through its own metabolic process to create glucose. So this is the actual stage part here, where it actually leaves the Calvin cycle to create a glucose molecule. So remember, you need a total of 2 G3Ps to make a glucose, and only only one G3P will leave if there's a total of 6 glyceraldehyde 3-phosphates that are present. Okay? So now we're going to be looking, so that was step 2 or stage 2. Now we're going to be looking at stage 3. So this is the part where we're going to go back into regenerating and creating our UBP, because again, this is a cycle or a cyclic type of cycle, so we need to start and end with the same product. So RUBP is necessary to keep the Calvin cycle running, so it must be regenerated by the end of the cycle. The remaining 5 PGAL molecules and 3 ATPs in a series of complex reactions to regenerate RUBP. So again, looking at the specific image here, if there was a total of 6 glyceraldehyde 3-phosphate and we lost 1 that's going to later move on to make glucose, then as we continue the cycle into phase 3, we only end up with a total of 5 G3Ps. And essentially, the ratio here gets a little wonky because it's not a nice 1-to-1 ratio that we saw here in phase 2. However, there are 3 ATP molecules that are required or needed to allow for G3P to be phosphorylated to create our RUBP. So and again, without recycling the RUBP, then we can't keep going and we can't keep using this Calvin cycle. So 1 molecule of PGAL requires 3 carbon dioxide molecules. To make 1 molecule of glucose, it requires 2 PGAL molecules. So that's why you need a total of 6 carbon dioxide molecules to actually allow for the creation of glucose. So therefore, 1 glucose molecule requires 6 carbon dioxide, 18 ATP, and 12 NADPH. So what does that mean? If you look back at this image here, simply you would have to multiply everything by 2. So you would need a total of 6 carbon dioxide, which means 12 phosphoglycerate, 12 1,3-bis phosphoglycerate, 12 glyceraldehyde 3-phosphate, which means that you're going to lose 2 of these G3Ps. That's going to go on to create 1 full glucose molecule. Then as you enter into stage 3, you end up with a total of 10 G3Ps, which means that you would need a total of 6 ATP to allow for a total of 6 RUBP. Okay, and that means that these numbers also double over here. You need a total of 12 ATP and a total of 12 NADPH. So where did we get these numbers from? The amount that's required to make one glucose molecule. So again, we need a total of 6 carbon dioxide. We see here that if you were to multiply this by twice, because you're only using 3 carbon dioxide, so if it was a total of 6, then you would need 12 NADPH. You would need 12 ATPs that's in this process over here, and then you would also need a total of 6 ATPs in the third phase over here, which makes up for a total of 18 ATP that's required to make only one glucose molecule. So a lot is needed in order to create just one glucose molecule. So I'm going to play this video for you guys here.
You're facing a giant bowl of energy-packed carbon crunchies. 1 spoonful, 2, 3. Soon, you're powered up by the energy surge that comes from your meal. But how did that energy get into your bowl? Energy exists in the form of sugars made by the plant your cereal came from, like wheat or corn. As you can see, carbon is the chemical backbone, and plants get their fix of it in the form of carbon dioxide, CO2, from the air that we all breathe. But how does a plant's energy factory, housed in the stroma of the chloroplast, turn a 1-carbon gas like CO2 into a 6-carbon solid like glucose? If you're thinking photosynthesis, you're right. But photosynthesis is divided into 2 steps. The first, which stores energy from the sun in the form of adenosine triphosphate, or ATP. And the second, the Calvin cycle, that captures carbon and turns it into sugar. This second phase represents one of nature's most sustainable production lines. And so with that, welcome to the world's most minuscule factory. The starting materials? A mix of CO2 molecules from the air and pre-assembled molecules called ribulose bisphosphate, or RUBP, each containing 5 carbons. The initiator? An industrious enzyme named Rubisco that welds 1 carbon atom from a CO2 molecule with the RUBP chain to build an initial 6-carbon sequence. That rapidly splits into 2 shorter chains containing 3 carbons each and called phosphoglycerates, or PGAs for short. Enter ATP, and another chemical called nicotinamide adenine dinucleotide phosphate, or just NADPH. ATP, working like a lubricant, delivers energy, while NADPH affixes 1 hydrogen to each of the PGA chains, changing them into molecules called glyceraldehyde 3-phosphates, or G3Ps. Glucose needs 6 carbons to form, made from 2 molecules of G3P, which incidentally have 6 carbons between them. So sugar has just been manufactured, right? Not quite. The Calvin cycle works like a sustainable production line, meaning that those original RUBPs that kicked things off at the start need to be recreated by reusing materials within the cycle now. But each RUBP needs 5 carbons, and manufacturing glucose takes a whole 6. Something doesn't add up. The answer lies in one phenomenal fact. While we've been focusing on this single production line, 5 others have been happening at the same time. With 6 conveyor belts moving in unison, there isn't just 1 carbon that gets soldered to 1 RUBP chain, but 6 carbons soldered to 6 RUBPs. That creates 12 G3P chains instead of just 2, meaning that altogether 36 carbons exist, the precise number needed to manufacture sugar and rebuild those RUBPs. Of the 12 G3Ps pooled together, 2 are siphoned off to form that energy-rich 6-carbon glucose chain, the one fueling you via your breakfast. Success! But back on the manufacturing line, the byproducts of this sugar production are swiftly assembled to recreate those 6 RUBPs. That requires 30 carbons, the exact number contained by the remaining 10 G3Ps. Now, a molecular mix and match occurs. 2 of the G3Ps are welded together, forming a 6-carbon sequence. adding a third G3P, a 9-carbon chain is built. The first RUBP, made up of 5 carbons, is cast from this, leaving 4 carbons behind. But there's no wastage here. Those are soldered to a 4th G3P molecule, making a 7-carbon chain. Added to a 5th G3P molecule, a 10-carbon chain is created. Enough now to craft 2 more RUBPs. With 3 full RUBPs recreated from 5 of the 10 G3Ps, simply duplicating this process will renew the 6 RUBP chains needed to restart the cycle again. So the Calvin cycle generates the precise number of elements and processes required to keep this biochemical production line turning endlessly. And it's just one of the hundreds of cycles present in nature. Why so many? Because if biological production processes were linear, they wouldn't be nearly as efficient or successful at using energy to manufacture the materials that nature relies upon, like sugar. Cycles create vital feedback loops that repeatedly reuse and rebuild ingredients, crafting as much as possible out of the planet's available resources. Such as that sugar, built using raw sunlight and carbon, converted in plant factories to become the energy that powers you and keeps the cycles revolving in your own life.
So essentially that's the whole process of the Calvin cycle that you see here. So I mean, you don't have to worry about the whole process of it going back and forth and how it rebuilds the RUBP, but you do need to note that you need a total of, so again, let's say we're using 6 carbon dioxide this time and you end up with 10 G3Ps, you would need a total of 6 ATP that are present to allow it to create 6 RUBP. Okay, so then you understanding the ratio and the number that's needed is important. And also what's important to note is all of the different names. So like what you've done in your cellular respiration cycle, you need to know all of the different names and also the importance of how big these different molecules are. So an overview of the photosynthesis reaction. So a lot more complicated than we had learned when we were younger. Now we're getting to the nitty-gritty detail of it. So the first part is a light reaction where it actually depends on the light itself, the photon of energy to kickstart off this process, where water is needed in order to create it, and it creates oxygen at the end of it. Then the products from the light reaction, like NADPH and ATP, are needed in the Calvin cycle along with carbon dioxide to finally produce our glucose. And then again, the products that were used, NADP plus and ADP, will then cycle back into our light reaction so that it can be converted back into ATP and NADH to be used in the Calvin cycle process itself. So here's just an overview. So light-dependent reaction produces ATP and NADPH, requires direct involvement of light, takes place in the thylakoid of the chloroplast. Light-independent reaction, carbon dioxide molecules are assembled into glucose molecules, does not require direct involvement of the light, and it takes place in the stroma of the chloroplast. Here's the homework that you need to complete for this part of the lecture. So please make sure you read pages 166 and complete the homework questions 167 to complete homework questions on page 167, which is numbers 13 to 18.